Good afternoon. Thank you very much for coming along today, or as I said earlier, this morning or this evening. And we're proud to welcome you to what we hope will be the first in a series of presentations and talks which are aimed particularly at perhaps PhD researchers or postdocs or those early in their career. And we want to do these topics on subject matter that are of relevance to you. So please do contact us here at NBIC by our contact details, which we'll be sending out or you may already have. And this first talk, I'm pleased to say, is one which we were specifically asked for and which we have an organisation who've come along to do. And I'll hand over to them in a moment. If you have any questions during this event, then please do use the Q&A box, which you should see at the bottom of your screen. And we will pick all of these up at the end. If you do happen to put them in the chat box, then we will still capture those, but it's easier to organise if they're in the Q&A box. So without further ado, I just wanted to say thank you again and hand over now to Jennifer Mackay and Dana Walsh from CosmosID, who are going to talk to us on utilising metagenomics in early to late stage R&D. So I'm going to hand over to Jennifer and Dana to take this further. Thank you.
Great. Let me just share my slides. Is the right screen sharing for you all? Can you see the correct?
Yes, it is. You're fine. Thank you very much.
Awesome. So thank you for having us. Great to be involved. Great to be invited by NBIC. So the talk today is really almost an intro to metagenomics, as Mark said, for PhDs, postdocs in the NBIC community. So what I'm going to talk through, I'll start. So lovely to meet you all. I'm Jen. I work as the academia lead within the business development team here at CosmosID. What I'm going to do is walk you through a standard metagenomic workflow to kind of set the context and to introduce CosmosID. So for anyone new to this, it should kind of set the foundations for what a metagenomic study entails. I'm then going to pass over to Dana. So she's our microbiome scientist, has much more expertise than I do, and is going to go into more granular detail through examples of analyses, and we'll leave plenty of time as well at the end for any questions. So to start with a little bit of context, metagenomic studies, they're just incredibly complex in nature. So at every single step of the workflow, there is the opportunity for error or for bias of some form. This can start with the sample type being challenging to deal with. You have biases through sample collection, sample processing, all the way through to the industry having been very reliant on inaccurate and incomplete public data sets for comparison. So to introduce CosmosID, we were founded back in 2008 by Professor Rita Colwell over in the US. Rita's a really well-known microbiologist, and she did a lot of work in biothreat detection. So Rita was really excited by the prospect of using NGS data to kind of bypass the traditional microbiology techniques. So her goal was to put together a suite of computational tools with the end goal of achieving strain level resolution from sort of any complex sample. So we started with the analysis portion of the workflow. What then happened was we realized that we could really add value to all stages in the workflow using our microbiome and our genomic expertise to overcome a lot of the industry bottlenecks that are shown on screen. So essentially we diversified, added on a wet lab to our dry lab work so that we could generate good quality NGS data to feed into our pipeline. So that's a bit of a whistle-stop tour on us. Where we are today, we're in essence a microbial genomics end-to-end partner, and we do support all stages of the metagenomic workflow. This is really what I'm going to talk through today, but from more of a conceptual point of view, as opposed to just focusing on us at CosmosID. So here you have your standard workflow for metagenomics. As mentioned, at each step here, it's very easy to insert error. So for that reason, you really need to be meticulous in the planning, in the standardization, and in the optimization of every single step. just as a bit of a side note, we do work across various industries. I know that biofilms is quite a varied heading. So we're not siloed into one specific area. The majority of work that we do is in the microbiome field, but we also work in infectious disease, diagnostics, really anything with a microbial component. And then the application of this is, of course, very, very broad. So hopefully there is something for everyone in here. And as I say, I'll now walk through a workflow and kind of touch upon overcoming industry bottlenecks. So starting with the study design, we of course begin with sample collection, something that can often be overlooked. It's perhaps a little bit boring, but is really incredibly important in the success of any microbiome study. So there are various papers that really look at how different collection methods will impact the results. The overriding theme here is that you just need to make sure the protocol is exactly the same per sample for the results to be truly comparable. So standardization is just so key here. When the specifics of the method itself are most important is really when you consider sample types that are more challenging to deal with. So those perhaps with a low microbial biomass, a lot of host DNA. On that theme, the example shown here is for skin samples. Some considerations that you need to think about will include the length of time spent swabbing, where you're actually swabbing, whether you're pre-moistening your swab, and crucially, the DNA extraction kit that you use at the other side. These will all have a large impact. There are then a few options as to how you go about storing your sample. So it's very rare anyone's going to be directly processing the sample for DNA analysis straight away. So you can either use a preservation buffer or you can freeze the sample, of course. Now, there are pros and cons to each. If we're talking the gold standard for microbial analysis, it's always going to be fresh frozen. This is really so there's as little manipulation to the microbes in the sample as possible. And it can really help to improve DNA yield for more challenging sample types. The major downside here is, of course, that you need to have -80 freezers, and not everyone's going to have that. So there are also preservation buffers available that are very commonly used, much more convenient, and they do allow for ambient storage anywhere from a month to 2 years, depending on the buffer that you're using. The main downside here is that you are going to be manipulating the microbiome to some degree, and the DNA yield can be negatively affected as you're in essence going to be diluting the sample. So this may be more problematic for samples that are known to already have a low microbial biomass. What we do to support this area is we can put together fully assembled kits with all of the necessary components. This is often used by our partners in the clinical space. So we ship out fully assembled patient-facing kits. This helps with the collection protocol, making sure that it's totally standardized. We do also develop kits on a more custom basis. So I'm working with a group in Turkey on a mucilage project. And there we're able to help develop the best protocol on a custom basis alongside them. So now we have your sample, we obviously move on to the lab work. The first important point to consider here is that any lab workflow should be optimized towards your specific sample type. So for example, using the correct extraction kits, adding any necessary pre-extraction steps. So we've got experience in a really wide range, about over 20, and we've worked alongside our partners to optimize different workflows, crucially making sure that you're optimizing for DNA yield or for purity in low biomass samples in particular. Now, just briefly, our lab is based in Germantown in the US. We're also soon opening a sequencing facility in Sweden, which is very exciting. Because we're a commercial lab, there are various processes that we have in place for standardization and for quality. First, samples will only ever make their way through a one-way lab system. We also use high throughput robotics to minimize error in the lab prep and the lab work. And our processes have been CLIA certified and we're GCP compliant, meaning we can support clinical studies. Now, even if you have these sorts of measures and protocols in place to reduce contamination, given we're dealing with microbes, it can be tricky, and adding in controls is really essential. So our personal procedure is that we do a series of 3 controls for every run, so negative, positive, and spike in. Without going into really any form of granular detail here, in short, if contamination does occur, at least we can identify where it came from and normalize or rerun as needed. So lab practices aside, when we get to the actual sequencing, there are a couple of options. I'm sure the group here will be more than familiar with all of these, but typically for metagenomic studies, you're looking at either amplicon sequencing or whole genome. Now, with amplicon, you're of course only looking at one specific gene or even specific hypervariable regions from one gene that are then taxonomically informative. Shotgun metagenomics, on the other side, is a whole genome approach. So you sequence all of the DNA, host and microbial, and then following this, you bioinformatically sort of tease apart the sample. Now, both of these approaches, you would then often refer to reference databases as part of your analyses. Based on your research question, different research methods are going to be more appropriate. And as you can see, there are pros and cons to each. So for amplicon sequencing, this works best for samples with high background or host interference, such as tissue samples or blood, which has a lot of host DNA to cut through. You can cut through that by just analyzing the 16S genes of bacteria, for example. Major downside here, you only truly get genus level insight with confidence. And there's also an amplification bias that's associated with this method. Then there's whole genome, so shotgun. We've supported a lot of academics in making the switch to shotgun metagenomics, particularly shallow shotguns starting at sort of 3 million reads per sample. Now, shotgun in general will give you multi-kingdom ID, species and strain level detail, and you also don't get that false positive rate that you would from 16S. If you're really interested in deeper characterization, shotgun does also open up a lot of other doors. Because you're sequencing all of the DNA, you're not just limited to taxonomic, but you open up functional analysis. So looking at pathways and genes. Fundamentally, though, it's really going to be your study question that will dictate what method makes the most sense. For shotgun, the first consideration is going to be what sequencing depth you target. So essentially how much coverage you need of the metagenome. So you can see here some examples we have of human samples we've dealt with. The composition of your sample alone will influence how you might want to sequence that sample. So if I bring my mouse up here, you can see stool samples down here. They have high diversity, almost no host DNA at all. That's why we can do shallow shotgun on these samples. On the flip side, where you've got the vaginal microbiome up here or skin samples, for example, there's barely any microbial biomass at all. There's a lot of host DNA to cut through. So because of that, you're going to need to go deep enough to cut through the human reads and get enough coverage of the microbes. So something like 20 million reads may be a more appropriate starting point. So sample type alone will help you dictate the sequencing depth that may be required. And you would then also look at your study question to get a feel for how sensitive the assay needs to be. To further this, trying to answer the question of, what is the correct sequencing depth, it's pretty complex, as in reality, there's not a single right answer. Different depths are going to give you different information. What we've done internally is we've done a lot of benchmarking studies in-house to try and support this question. So here we have increasing sequencing depth alongside alpha diversities. We've got richness here. Given there's clearly a strong plateau both for skin and for stool, perhaps for your study, it's really not a case of the deeper the better. For example, you may go into a study thinking we want to do 20 million reads per sample, but if you can answer your study question effectively at 12, you'll really be able to cost optimize your project. To further that a little bit, in some additional benchmarking, we actually found that there was a correlation of about 96% for microbial profiles of moderate, so 9 million reads here, and very deep, so 90 million reads, and this is stool samples we're looking at. Really what this means is that if you're only interested in general microbiome profiling, you don't need to sequence ultra deep. However, the caveat there is if you are interested in that bottom 4%, that rare low abundant microbe or function, then increasing the sequencing depth from 9 to 90 increases the sensitivity tenfold. And so this is really why considering the sequencing depth is important. How sensitive do you need the assay to be? In what abundance do you expect what you're interested in to be? These are the sorts of questions you should ask when you're embarking on these studies, and you can then influence the limit of detection through increasing the sequencing depth. So now we have sequenced your sample, we of course move on to the bioinformatics. For the purpose of this webinar, I'm going to focus on the CosmosID approach for shotgun. And our real goal, our overriding goal, I guess, is to access strain level and certainly species level resolution. Now, why that's even important is that the strain is really considered to be the clinically informative and actionable unit. There are a few common examples of this. So E. coli is a really nice one. You've obviously got a commensal strain, you've got a pathogenic strain. There's also an example with lactobacillus here where different strains give you yogurt, cheese, wine. All of them are absolutely great, don't get me wrong, but they're obviously very different. So when we're considering this in the context of R&D, getting to the strain level is really where you're going to see the difference in functionality. Now, taking it one step further, why is that actually important? So this is a skin case study here, but it's comparable for other sample types. The paper is looking at psoriasis versus healthy controls. It's done by Nicholas Sagata's group, and they did see an overabundance in Staphylococcus in psoriasis patients. Now, we also ran this data through our pipeline and we kind of zoomed in, and there is so much more variation than that to be seen. So within Staphylococcus, there's a lower diversity in psoriasis patients. Different strains are also present. So most of the variation that you're seeing is actually at the species and the subspecies level. We did a kind of whole webinar on this. So if you are interested, feel free to head there and check it out. To kind of take it a step back, perhaps using 16S, you may not have seen hugely different results in your study, but with whole genome, you can see that granular detail where changes have occurred. So how we can achieve strain level resolution actually starts firstly with our database. I mentioned one bottleneck previously is the kind of inaccurate public data sets in the industry. So the way that we work is we curate genomes and then phylogenetically structure every single genome in our database. So what that allows us to first do is identify microbes that have been misclassified. So we do that based on the surrounding neighbors. So what you're getting is a higher quality comparative data set for your analyses. The phylogenetic structure, most importantly though, enables us to identify taxonomically informative k-mers or markers at different levels of the phylogenetic tree. And that's how our algorithm works. So our algorithm uses what's known as a k-mer-based approach as opposed to an assembly-based approach. And without going into any form of detail here, in a k-mer-based approach, the algorithm will look for a combination of shared and unique markers across the phylogenetic tree. And we will then assign a classification based on the distribution of this. So that's how we can access strain level resolution. We have benchmarked this against various other tools. You can see on screen, we had significantly improved recall and precision, but rather than bombard you with any information on this, I'll of course share the slides with you, and you're more than welcome to read the the blog and publication here in your own time. So as well as taxonomic information, the good thing about shotgun data is that you then have the option to look at functions. So here's an overview of our functional pipeline. The output here is that you get both meta-site pathways and Go terms with the one pipeline. You can then start to kind of correlate your taxonomic info with potential functions. What I haven't talked through at all here is that you can, of course, do this same workflow with RNA, so metatranscriptomics. This often ties in very nicely to functional analysis, as rather than an inferred function from DNA, you're getting more of an active view on what's present through the RNA in the sample. Now, the final hurdle, once you have your raw reads, you've run them through your bioinformatic pipelines, is the analysis. And before I hand over to Dana, who's really going to talk you through a couple of case studies and bring this all together, I wanted to show you briefly the CosmosID Hub. So metagenomic analysis can be incredibly complex. What we've done is we've automated our pipelines and made them accessible online via the CosmosID Hub. It's really very user-friendly, so you don't need a bioinformatics background to get the most out of it at all. Because it is very fast and we've got command line functionality, bioinformaticians themselves do find it useful, but within the hub, you've got access to multi-kingdom ID, as well as AMR genes, virulence factors, meta-site pathways, Go terms, and you've got unlimited comparative analysis available to you as well. So all of the bread and butter analysis, some of which Dana will walk through, so PCA for similarity, alpha, beta diversity, LEFC biomarker analysis. And what makes the hub really popular, particularly amongst academics, is that it makes microbiome data and visualizations and indexes and charts accessible to all researchers. So you can really spend more time interpreting the data, getting ready for publication, as opposed to generating simply visualizations. Now, Dana's going to show some of the outputs of this, as I mentioned, and if we do have time and if there is interest later, I'd be more than happy to walk you through a quick demo of the hub as well. So there are, of course, many advanced stats that are perhaps outside the remit of the hub for now, but if you do ever need advanced support or would like to talk through something in more detail, we've got a team available, and I'm also happy to share any example reports on what can be done. Now, this brings me up to the end of my section. I'm going to stop sharing my screen and pass over to Dana, who will walk you through some microbiome analysis.
Great. Well, good afternoon and good morning and good evening to everyone. Happy to be here and talk to you today. So let me share my screen here quickly. Or it is sharing. Play. All right.
Thanks, Dana, that's good. Yep, that's good.
Great. Okay. Yeah. So as Jen said, I'm going to walk you through an example, really, data set of how we do the analysis. So the first portion will be, most of it actually will be kind of our standard outputs that you would get with, if you did your analysis through CosmosID and also got analysis help back, these would be the standard outputs you would get, and also what you could generate yourself on the hub if you wanted to. Oops. Okay. So before we jumped into it, I wanted to give a little overview about some challenges and considerations we need to take into account with microbiome analysis. So it's challenging because it's big data. So there are some different things you have to think about. So as Jen was saying, you can do 16S sequencing, you can do whole genome sequencing. This data set is whole genome sequencing, and that's really our bread and butter at CosmosID. It's an untargeted approach. So again, you're sequencing the whole genome versus just that 16S gene. So that allows you to identify all the bacterial genes present. So then you get an indication of the functions, like what are the microbial communities doing, which can be really helpful to enhance your interpretation in addition to the taxonomic identification. And of course, we're also getting the taxonomic IDs here. It is more accurate than 16S is going to be. I'm not going to go into 16S here, but just briefly, there's amplification bias associated with 16S sequencing, as well as bias in what each of your primer sets are actually targeting and amplifying, and you don't have that with whole genome sequencing, so that gets rid of that whole problem. Some other issues, again, I said it's big data, so it can be computationally intensive to analyze this data. It's sparse, meaning there are a lot of zeros in the data set. So you may have, say, in a human gut sample, maybe we have 300 unique species or more. Well, not every sample is going to have all those taxa detected. So you end up with a lot of zeros in the data set. And that also contributes to the fact that your data isn't going to be normally distributed. So you can't use your normal parametric statistical tests to analyze this data. We have to use non-parametric methods and other specialty methods that have been developed for this analysis specifically. So the general approach in the field, and also our approach at CosmosID, is to first apply some diversity metrics that are taken from ecology to this data. So alpha and beta diversity specifically. So alpha diversity will tell you what number of bacteria are there and how many of them. So what number of different taxa really within each individual sample. Beta diversity kind of expands on this and is a way to look at how similar are the microbial communities to each other, similar or dissimilar. Then the next question you might have is, well, what taxa specifically are important to your research question, whether that's a disease or some type of environmental question, location, whatever that may be. But we can use differential abundance methods to address that. So we've been using a tool called LEFSA at CosmosID, and I'm going to explain that in more detail in a couple slides here. And so another thing we can do that I have listed under custom or advanced, and I have it listed this way because LEFSA and alpha and beta diversity are all included in our standard output. But if you are interested in diving deeper into the data, we can do some more custom things such as machine learning. So I'm going to show you an example of how we can use machine learning and the microbiome to make predictions about disease outcome based on the microbiome composition. Some other examples are network models, so you can actually do some correlations and look at the relationships between the taxa, their functions, and maybe disease or other patient metadata, immune markers, for example, or other omics data sets, metabolomics, metatranscriptomics, for example. So this data set I'm going to be telling you about today is an example set from Crohn's disease. And just briefly, Crohn's disease, along with ulcerative colitis, is considered an inflammatory bowel disease. So Crohn's can affect the entire digestive tract, this entire area that I'm showing you here, with these skip lesions. And the cause isn't known, but there's a variety of different factors that are known to contribute to it, including genetics, including environmental factors, you know, diet, where you live, chemical exposure. There are immune factors and also microbiome factors that have been associated. And I'm listing several papers here with specific associations that have been found with Crohn's disease. A general finding in disease with the gut microbiome is a loss of diversity associated with disease. And we see this in Crohn's too. So as compared to healthy, the healthy gut microbiome tends to have high diversity. All right, so since this is whole genome sequencing data, I'm going to show you analysis of both the taxonomic data as well as the functional data. So you get an idea of the richness of the data we can get out of this method. So one of the first things we do is to look at a stacked bar plot. So this kind of gives you a visual overview of what taxa are present and whether or not there are obvious differences between your groups. The way this is plotted is Crohn's on one side, healthy on the other. And it's split by female on your left side and male on your right side. And you'll note here that we're only showing you the top 25 most abundant taxa. And I mean, that's just kind of a logistics thing because we can't show you all 300 taxa and be able to discriminate between the colors. So what you'll note here is that so both of these healthy bars in females and males look similar color-wise. So we have dominance by Bacteroides vulgaris. And I should mention this is at the species level. We have a lot of other Bacteroides species here that are dominating in our healthy groups, which we expect. So the healthy gut does tend to be dominated by Bacteroides. In Crohn's disease, however, you'll notice we aren't seeing as much dominance by these Bacteroides species. Instead, this purple is Bifidobacterium longum. The pink, I believe, is Bifidobacterium adolescentis. And then in this orange, we have a Lactobacillus amylovoris. So those species aren't necessarily pathogens, but they don't tend to be found as dominant species in the healthy gut. So this might be an indicator of dysbiosis in the Crohn's disease gut microbiome. So that's just an observation, and obviously we want to apply some statistics to this. So again, as I mentioned, we can look at our kind of general overview of what does the community look like by starting with alpha diversity. And I'm including this figure to show you, to explain alpha diversity really. So alpha diversity incorporates richness and evenness of your community. So richness essentially is the number of different taxa, and evenness is how many of those taxa are present. So in this community on your far right, it has high richness and high evenness because it has a lot of different fruits, but there's 2 of each fruits. So that's pretty even, versus this community right next to it has high richness, again, because a lot of different types of fruits, but low evenness because this is dominated by apples. There are a lot of apples here. So there are several different metrics we can use to look at alpha diversity. Today, I am going to show you the Shannon diversity metric. So I like Shannon diversity personally because it's a pretty balanced and robust metric. So it takes into account richness and evenness. And it's not, some of the other metrics can be dominated if there's lots of less abundant taxa, or I should say skewed, not dominated, or it can be skewed by dominant taxa, or some metrics only look at the number of different taxa. Shannon is a pretty good balance between those. And what you're seeing here is the median Shannon diversity index, indicated by the line in the middle of the box plot. It's showing you the 3rd and 1st quartile ranges here as well. But we do see a significant increase in alpha diversity in our healthy subjects as compared to the Crohn's disease subjects. And again, this is what we see in other diseases as well as previous Crohn's disease studies. And that decrease in alpha diversity might indicate some sort of dysbiosis going on here. So the next metric is beta diversity. And this figure is kind of tying together alpha and beta diversity. So it's showing you in these different ponds here, we have different numbers as well as types of fish. So that's alpha diversity when we're looking at these individually. Beta diversity now is we want to compare how similar these ponds are to each other. Oops, and we can do that. Again, there are different metrics that can be used to assess beta diversity. We tend to use the Bray-Curtis method. Again, it tends to be a more balanced, robust method. It takes into account the number of different taxa as well as their relative abundance. So in this plot here, each dot represents a microbial community from an individual patient sample. And the closer they are to one another, the more similar they're going to be. So the way this is colored is we have purple and red is male and female healthy, and then blue and green over here is male and female Crohn's. And you can see pretty clearly, and it's kind of circled in this plot, that the Crohn's and the healthy communities cluster distinctly away from each other. You're not seeing overlap between them, which would indicate that they're pretty different. You are seeing overlap based on gender within Crohn's versus healthy. So that would indicate that the disease really is the driver of the differences we're seeing here and not gender. All right, so we can also look at heat maps here, and I like heat maps because they give you a quick overview of what are the most abundant taxa in your data set per sample. So we have taxa here, and then in the columns are individual samples. I don't have them labeled here, but that's what they are. And then I've colored sex across the top, and underneath that, Crohn's in orange and healthy in this salmon color. And you can see pretty clearly that there are taxa present in Crohn's that aren't present in healthy and vice versa. So these communities are pretty distinct. We also ran a Kruskal-Wallis chi-squared test. So this is a non-parametric, essentially t-test to look at whether the relative abundance of these taxa are significantly different between the groups. And we also did a adjusted p-test, a false discovery rate here, since we're doing multiple tests at once. But what I'm showing you here are the taxa that were significantly different between healthy versus Crohn's. And we're seeing a lot of species that we would expect in a healthy gut: Coprococcus, Bacteroides, Allostypes to some extent, but then Lactobacillus amylivorus again is showing up here. So the next question then is, which of these are really distinct between healthy versus Crohn's? And we can talk about lefse, which is a delicious flatbread, and I like it with cinnamon and butter. Actually, it's not that much fun in this context. So lefse in this context is actually a statistical test, not bread, unfortunately. It was designed by the Huttenhauer lab. It stands for Linear Discriminant Analysis Effect Size. So its purpose is to find taxa that are discriminatory between two groups. It can do more than two groups, but also to tell you whether those are biologically meaningful or consistent. So it does that by using these three statistical tests. And I'm not going to go into the details on how they work, but this is the output from lefse. So it gives you this chart back out. And in this case, in green, it's showing us taxa that are significantly discriminatory of healthy subjects. And then in Crohn's, in red, we have taxa that are significantly discriminatory of Crohn's disease. And they're discriminatory in comparison to each other. So if we look specifically at the bacteria that are discriminatory and healthy, we're getting, again, a lot of Bacteroides in here, which has been a common theme. Again, we expect Bacteroides to be prevalent and dominant in healthy samples. In the Crohn's disease samples, though, we're seeing things like Bifidobacterium longum, Lactobacillus amylivorus again, some Ruminococcus, some Coprococcus. So these are also found in the healthy gut, so they're not traditionally pathogens. However, they also aren't typically found as dominant in the gut. So this might indicate that there is some dysbiosis of the Crohn's disease gut microbiome going on here that could be contributing to the pathology we see in the gut. Okay, so we've gone over the taxonomic data. I will take you through the functional data, and this will be a little bit faster since we've already defined our diversity metrics. So again, with the functional data, we use a similar approach. Stacked bars are a great way to get a sense of, are there clear differences between your groups? And generally with functional, there isn't, just because there tend to be so many terms that are identified. And when we only show the top 25 here, it's difficult to see that there are clear differences. However, we can apply essentially our beta diversity metric. So this is, it's clustering your samples again based on how similar are the functions now between our groups. And again, here you see in red, the healthy, and blue Crohn's, and they do cluster distinctly from each other. And again, this is significant. So this would tell us that these two groups are significantly different, also based on the functions we found within the data. And again, we can apply LEFSA here because, of course, your next question will be, well, which of these functions are important or significant in discriminating these two groups? And just to go through this briefly, in healthy, we're seeing pathways that are more involved in metabolism and nucleotide synthesis. So these might be things that are more associated with just normal homeostasis of the cell, normal maintenance. In the Crohn's disease, however, we're seeing increases in amino acid synthesis pathways, as well as this UDP N-acetyl D-glucosamine biosynthesis. Wow, I said it too fast. So this is interesting because it's a precursor to peptidoglycan found in bacterial cell walls, obviously. So perhaps this indicates that there is growth of new cells here in the Crohn's disease bacteria. Whether or not that contributes to the pathogenesis, we can't say from this data specifically, but we can do further analyses to kind of get at that. So this data I've just shown you is what you would get in a standard analysis output from us. What I'm about to show you next would be considered custom. So that's something you'd work more one-on-one with us to do and to answer your specific questions for your specific groups. So what I've done here is to use a machine learning method called Baruda to predict taxa that, to find taxa that are predictive of Crohn's versus healthy. So I want to give you a brief overview of what Baruda is and how it works. If you're not familiar with machine learning, essentially Baruda is a god of the forest. So Baruda is a wrapper around a random forest algorithm. And the random forest algorithm itself is made up of decision trees. That's the most basic unit. So a decision tree, what it does is to find the best parameters in your data that discriminate between them. So between healthy and Crohn's in our case, and we're looking at what taxa are most discriminative. So in this example here, we're looking at these numbers that are different colors and different numbers. So what's most discriminative here? Well, color. So it's going to split first on color. And then this blue group doesn't split any further because there are no more differences here. However, we can further split this other group either based on the underline or the number itself. And here it shows underlined. And the question then might be, well, is splitting by underline or number better? And then that's where random forest comes in, because random forest does this iteratively many different times. And that helps you get a more robust prediction of what parameter is the best for dividing your data. And essentially what random forest will do is rank your parameters based on the importance of them in doing this prediction. But with random forest by itself, there's not a way to establish a cutoff or a threshold. It's kind of arbitrary. You just get to choose. And that's why I use Baruda, because what Baruda does is bring more statistical robustness into choosing where that threshold lies and where you cut off, okay, these are my important taxa in making this decision. So this is the output from Baruda based on the Crohn's data. So I'm showing you here in green only the taxa that Baruda identified as important in splitting or identifying Crohn's from healthy. So there were other taxa that I'm not showing you here that it rejected. It said they weren't important. You'll notice though that there's this group, there's about 14 here that have higher importance than these other taxa. And I'm showing you them here in this chart along with the importance, which is around 3. And I just want to note that the importance number itself isn't biologically meaningful. It's specific to your data set. It's just a way that Baruda has of ranking your variables. So it tells you what's most important in your data set. So what I did next was to take those 14 taxa and run an ROC curve analysis. So the purpose of the ROC curve analysis is to tell us how good is our test at differentiating healthy from Crohn's. So are those taxa actually predictive? And I got an area under the curve of 100%, which is like the best you can get, which is great. If you remember back to that heat map, there were definitely bacteria that were only present in Crohn's and that were only present in healthy. And I think this is why we're getting the AUC of 100%, because those top important taxa, if you look at my relative abundance plot over here, I'm showing you, so they're the same taxa in each of these across the bottom here. So actinomyces in both cases here, Faecalibacterium in both cases here. And you can see pretty distinctly that there are taxa that are enriched in Crohn's only, and we have either very low or zero abundance of these other taxa. But those ones that we have the low abundance of in Crohn's are actually enriched in healthy. So we have pretty distinct sets of taxa that Baruta has found for us here that can differentiate Crohn's from healthy. So you could take this a step further and take these taxa, do some more testing, and perhaps develop them as a biomarker of Crohn's or differentiating between Crohn's versus healthy. Okay, so to tie this all up, I've walked you through the basic approach to microbiome analysis. I showed you taxonomic and functional data from a Crohn's disease data set, and we saw that both on the taxonomic and the functional side, we definitely see differences. We see dysbiosis in the Crohn's disease, and we see distinct taxa that might be predictive of Crohn's disease based on the results from Baruta. Again, something else that we could do with this analysis is to do some correlations and do a network analysis so we can see what are the relationships of these important taxa with other taxa in Crohn's. Are there helper taxa that might help these important taxa grow that maybe physicians would want to target for treatment? So further analysis that could help us maybe understand further the pathology of Crohn's and how the microbiome contributes to that. So that covers my end of the talk. I think we're running a little low on time. I don't know if we have time to do a hub demo. Oh, I also wanted to mention the figures I showed you from our standard analysis, you can also produce yourself in the hub.
Thanks, Dana. I think we probably should take the questions first and then see where we are for time. Thank you both very much for fascinating two presentations there. I'm just going to stop you sharing for a moment. And then, Will, I think we've got 3 or 4 questions that appeared in the chat. So
yes.
if you'd like to pass those on, please. Thank you.
Sure. So the first one that came in was during your talk, Jen, and it was asking why would shallow shotgun sequencing not be ideal for AMR genes if there is little to no host contamination?
Yeah, really good question. So the slide was probably a bit too black and white. It's not saying that shallow shotgun. So for shallow shotgun, we're really thinking around 3 million reads per sample. That's typically what we mean. So it's about 0.5 gigabases. For something like a pure microbial biofilm, it would probably work. It really depends entirely on the sample type. It's kind of a question of coverage based on the diversity and the biomass of the sample. So shallow shotgun won't be appropriate for all samples to achieve AMR output. You'd often want to go a little bit deeper just to make sure you're getting enough coverage. If there's something of interest specifically, I'm happy to pop my details in the chat and you can reach out to me and I can perhaps answer that in more detail. Is there anything you'd add to that, Dana?
Yeah, I just wanted to say for like stool, for example, like shallow shotgun is usually sufficient for identification of AMR genes. It's just the deeper you go, the more you're going to identify. So, you know, if there's something that's not there at a high abundance, you might not see it with shallow shotgun.
Yeah.
Next one's come in was asking about, have you considered using, I don't know if there's QIIME2, I don't know if there's a shortened version of that, for alpha and beta diversity, and they go on to mention that it has the capabilities of calculating and assessing all the metrics, sorry, that you were mentioning in your talk. I think this came in while you were talking, Dana.
Yep. So yeah, Chime was developed by Rob Knight's group, and I used it a long time ago. I know it's improved a lot since then. It's a good tool. I use R, for example, to do this. You can use Python. Like R and Python give us a little bit more flexibility in doing the analysis and making the figures and things. So we could use Chime, or if we wanted to, and I do believe for some of our pipelines we do use Chime in some contexts. So
yeah.
Yep.
And for the alpha and beta specifically, I mean, there are just various different packages that all do similar things, right? So when it comes down to it, they're just formulas for alpha and beta. You can kind of use what works best for you. Yeah.
Sure. The next question that came in, this again was during your talk, Dana, and it asked, do you account for compositionality?
Oh, yes, definitely. So that is a big factor, and I should have mentioned that. That's a big factor you have to take into account, since all of this data is compositional. So we essentially normalize to the overall read count of the data to take compositionality into account, and we use relative abundance.
One that's just come in saying one of the figures in your presentations was comparing Kraken-2 against the CosmosID and other taxonomizers. Was the higher CosmosID performance related to the CosmosID database or to the algorithm itself?
I think we can both probably add to this, but I would say it's likely a combination of both, in that both of them combined are going to allow us to achieve more sensitivity. That the algorithm's a very sensitive algorithm, but we've also developed, and I didn't go into this in any form of detail, but our bioinformaticians have also developed precision filters for our shotgun pipeline. So these can essentially be toggled on via the hub, and they alter the detection cutoff point, essentially, so you're eradicating all of the false positives. So some algorithms will be perhaps as sensitive or in the same realms of sensitivity, but they're not as precise because they don't have similar filtering capabilities. And then because we've got a curated database, it obviously means we've got a higher quality database to compare it to. So I don't say that it's one necessarily over the other, but the combination of the two. I can also, I will make sure to share the slides, and there are a few links that will direct you to that study and to the blog post and everything. So do feel free to read through it. Dana, would you add anything to that?
Yeah, well, I definitely agree with everything you said. I also would add that like our K-mer approach it's a unique algorithm, and it's also very accurate. Like that's how we get to the strain level so accurately. So I think that's another big factor in how our pipeline outperforms Kraken-2.
Great. One comment about process of how you could help out with a study. Came in saying, would you need CosmosID to be involved at the planning stage of a study in order to best carry out the full analysis?
So we're very, very flexible. I'd say the best approach is to be as collaborative as possible, just because it means that both parties are fully informed. Also, if we know what you're really looking to achieve, then we can support and advise you from the get-go. However, everything that we do is modular. So if you did only need support in one area, that's absolutely fine. If you've got things set up already and you just wanted us to come in at a certain stage, that's also okay. But typically, you know, it is good to be involved from the beginning.
Sure. I think that's all the questions we had in so far. Oh, hang on. Isn't the question? Never mind. I was reading the first section. It says this isn't a question, so don't worry about that. A good statement about some of the things we were talking about. Yes, so that's it from my end.
Thank you for the feedback as well to the speakers you've given. So with the couple of minutes we've got left, Jen, if you did want to just show your hub, and then we'll wrap up on time, please.
Yeah, absolutely. I'll give you a whirlwind tour of the CosmosID Hub in the next 5 minutes. So is my screen sharing for you?
Yeah.
Cool. So first of all, just a side note, anyone can create a free account on this. So it's just app.cosmosid.com, create a free account. There's some example data sets that you can have a play around with. I'm going to focus mainly on the comparative analysis because it's a little bit more exciting, but just to show you what a single sample looks like. If I take you into a skin sample here, basically what you've got, I'll talk you through it briefly from top to bottom. Here you've got your read depth. So this is 24 million reads. This is the number of organisms that have been found. And here, the filter that I mentioned, if I toggle that on, you notice that it drops quite considerably. So we've lost likely a lot of false positives or really low abundance calls there. You then have the organisms listed by relative abundance. We actually give you the match statistics to our algorithm as well. So this is the number of hits the algorithm's made against this strain. And then there's various charts and visualizations. So sunburst, bubble, collapsible, radial tree, donut, and stacked bar. You can also toggle between data sets. So obviously, if you've done shotgun, you'll have multi-kingdom open. You can look at fungi, for example. And then you've got your functions as well that I mentioned. So metasite pathways, Go terms available. That's just in the one sample though. If I switch over to a comparative analysis, what's quite nice is once all your samples are uploaded, you can splice and dice them in as many different ways as you want. So there's no limit to the number of comparative analyses you set up. This is one I put together last week. It's publicly available data from a paper that I just imported. So I've grouped together vaginal samples. We've got HPV infected versus healthy control. Initially, you have your matrix table, which you can bring back to the strain level as well. And there's search functionality here. You can actually add up to 24 cohorts. So it can be quite nice for longitudinal studies. And then you've got a lot of the visualizations that Dana showed. So your heat map here can cluster by cohort, just look for the top 10 taxa, for example. Stacked bar, you can aggregate these, use random colors, and you can pair this back if you wanted to zoom out a bit to take it back to genus level. Now we move on to the actual stats. So here we have PCA. You can see there's no real clustering here. We then move on to alpha and beta diversity. So Chow, Simpson, and Shannon are available, and you can test for significance here as well. Similarly, here you can see that it is significantly different for Chow and Simpson. Shannon, you'd need to hit all to view the p-value. And moving into beta as well, we offer Jackert and Bray Curtis, testing for significance with Permanova analysis too. And LEFSIT that Dana mentioned has just been added on the hub this year, which is really cool. So the first output here is that you've got just the table. So you've got the list of bacteria and the enriched cohorts. But there's also a bar chart view. So you can change the threshold, you can change the p-value, and you get a visualization of those bacteria that are associated with each cohort. So that's a real whistle-stop tour. Everything can be exported. So images, tables, supporting data, etc. And as I say, feel free to create a free account. All of these example data sets you'll be able to see. So there's quite a few. And I hope that was useful. If there's any questions whilst I'm on the hub, we have 1 minute.
There's one, Jen, which was from Mackay. Will it extract info from global databases as well, or does it require a separate uploading process?
Dana, you're nodding. Do you want to take that one?
Yes. Let's see. I guess by global database, global databases. So it'll pull, we have an SRA import tool, so that will pull from, yes, data that's available on NCBI. So it will pull from NCBI. I mean, if you can get from other databases, if you can get it into NCBI, then we could upload it. Otherwise, we could definitely work with you to upload or compare to something something else that's not available in NCBI.
Yeah.
Thank you both. Okay, well, thank you. That's been a wonderful session. Very educational for all of us, I think. So thank you all very much. I just want to express on behalf of everyone who's been here today, Dana and Jen, for your time and for your excellent presentations. So thank all of those of you who have attended today for your attendance. And if you do have any questions for either NBIC or to the speakers today, do just contact us at nbic@biofilms.ac.uk. But when you get access to the presentations, I think Dana's and Jen's emails will also be in there as well.
Yeah, yeah.
So thank you all again very much, and have a good day, or however much of it is left for you wherever you are based. So thanks again.
Thanks, everyone.
Thank you.
Thank you.